Hmm. Hello, hello. We are back with another episode of the show where I try to learn something by reading interesting scientific articles. And the people who watch this show will listen to me mispronounce many scientific words. And I'm sure there's entertainment value in that. And maybe they'll learn something too. So, this article is about ants eating their own molting fluid, which sounds pretty gross, and I'm excited to dive into it. And the reason we're reading this one is because I just watched the movie Bug's Life, which is all about ants. And I'd never seen it before, and the animation holds up. So listen to this video and then go watch Bug's Life. Sorry, let's get started. Colonies of eusocial insects are complex biological systems that consist of obligately interacting individuals working cooperatively towards common goals. And so, eusocial, I actually recently learned this word. That is means a society where you have like one queen that's doing all the reproduction. Colony function and fitness are highly dependent on the types and frequency of interactions between its members, including adults and various immature stages. In ants, for example, adults groom, transport, and feed larvae. One important type of social interaction is tropophylaxis, the sharing of liquid food in the form of social fluids. The sharing of liquid food in the form of social fluids. What is a social fluid? I'm scared to know. Either mouth to mouth or anus to mouth. Ugh. Tropophylaxis is widespread among eusocial insects and is common in many ant species, both between adults and between adults and larvae. William N. Wheeler, who coined the term, even suggested that tropophylaxis would be the key to understanding eusocial behavior in insects. Interesting. And this is a study by all of these individuals here, and hopefully they get into more detail about where this study was conducted as we read through it. However, because developmental stages co-occur in close contact inside the nest, different types of social interactions might be confounded or difficult to discern. Therefore, we cannot fully describe the principles of interactions between individuals and how each developmental stage contributes and benefits from the social fabric of the colony using observations of intact colonies alone. In their 1990 monograph, The Ants, Bert Holdobler and Edward O. Wilson noted that this subject remains largely unexplored, but that we can reasonably anticipate many surprising new discoveries, some of which may force changes in our thinking about colony organization. I feel like reading about insects is particularly interesting because I think maybe we relate to them a little bit as humans because we kind of live in a colony like structure. Like a city is kind of like a anthill or a beehive in a way. We thus took an approach inspired by reverse engineering to discover new social interactions by isolating individuals, identifying resulting phenotypes, experimentally rescuing these phenotypes, and finally verifying and studying the relevant phenomena in the colony context. We focus specifically on ant pupae because pupae have been regarded as passive members of the colony, and to our knowledge, no direct contributions of pupae to colony function have been described. We use the clonal radar ant Ocera biroi as our main model system before generalizing our findings to other ant species. The clonal radar ant. What does that mean? A queenless clonal ant in the genus Ocera. Native to Asia. Wow. So they don't have a queen. I did not know that there were ants that existed that didn't have queens. What the heck? They reproduce clonally, so they're all clones of each other? Wow. This is crazy. Thelitoki. Type of parthenogenesis in the absence of... Wow. This is interesting. I don't know if they're going to talk about this in the study, but these ants that they did this study on are clones of each other. A pupal social fluid hinders survival. To investigate whether and how ant pupae interact with other colony members, we developed a protocol to rear O. baroi, I wonder how you pronounce that, pupae outside the colony in social isolation by providing the abiotic requirements for development. So we're going to click on this and look at this figure here. Figure 1A. Incubator and pupal fluid trap. We isolated the pupae from the beginning of pupatation until eclosion as adults, and we found that they secrete a substantial amount of fluid towards the end of metamorphosis. So, eclosion is a word I do not recognize. The act of emerging from the pupal case or hatching from an egg. Eclosion. Cool. That's a good one to impress people if you raise chickens or any other egg-laying species. So this looks like the fluid that is coming from the pupa. So they remove the fluid daily. Secretion begins 6 days before eclosion. Shortly after, the pupae have melanized. Does that just mean they get darker? The secretion initially accumulates in the exuvial space and then exudes from the rectal invagination of the pupal case, forming a stereotypical droplets on the abdominal tip. Secretion continues until eclosion, and the droplets are often still present on the exuvia. In subsequent experiments, we therefore isolated pupae 9 days before eclosion and just before melanization. During this period, on average, 99.93% of the pupae secrete fluid. As long as we manually remove the fluid daily, our social isolation protocol yielded high survival rates, with 90% of the pupae eclosing. Exuvia, I think, is just referring to the part of their body. The cast off outer skin of an arthropod after a molt. Okay. Exuvia. Among the pupae that survived the eclosion, we observed a stereotypical pattern. Melanization always preceded secretion. 70% of the pupae secreted fluid continuously over the course of 5 days before eclosion. Figure 1c. No melanization and no visible pupil fluid. No melanization and no visible pupil fluid. Oh, melanization and visible pupil fluid. Melanization and visible pupil fluid. Okay, so it's just saying the majority. Okay. No, this is. Yeah, so these are outliers right here. This is just showing days before they emerge from their molt. Yeah, so this is at this point they're still lighter color, and at this point they're getting darker, and now they're excreting the pupil fluid. On average, and the average daily volume secreted increased over time up until eclosion. Through the period of secretion, we collected 23.2 microliters of fluid per biological replicate pupae per replicate, per biological replicate pupae per replicate, approximately corresponding to the population of a natural colony species. Okay. This large amount of fluid secreted by pupae in isolation and the high fraction of pupae secreting daily, figure 1e, contrasts with the fact that the fluid cannot be readily observed when pupae are kept inside the colony with adult ants. 1e left and right. Fraction of pupae in the colony. So this is an isolation. Yeah, so you can't. Okay, that's probably because the adult ants are eating the fluid. We therefore tested whether pupae secrete the fluid in a natural colony setting, and if so, what happens to it. We injected blue food dye into the exuvial space of melanized pupae and traced its distribution in the colony. For several days, we found blue staining in the digestive system of all adult ants that had spent 24 hours with injected pupae. Wow. By the end of the experiment, no pupae had died and all of them responded to tactile stimulation, showing that the adults did not acquire the dye by cannibalizing the pupae. Instead, this suggests that the adults consume the pupal fluid immediately as it's secreted, which is why the fluid does not accumulate and become visible in the colony context. Indeed, adult ants pile up the pupae and remain in prolonged physical contact with them, frequently touching the pupae with their mouth parts. Oh, they have a video. I don't know if I have access to this video. Oh, that's unfortunate. We then isolate the pupae and allow fluid to accumulate before adding adults. The adults were highly attracted to the fluid and rapidly consumed it, preventing extrusion of the secretion by occluding the gaster tip of the pupae. Reduced ant attendance compared to secreting controls. 1G, okay. Occluded. Wow, that's crazy. That's so cool that they can see what the ant eats just by like dyeing things. You can track it through a colony. When they tested whether removal of the secretion affects the fitness of the pupae, daily manual removal of secretion was sufficient to achieve high rates of pupal survival and eclosion in isolation, similar to the colony context in which pupae were in a nest with a group of adults. Figure 1H. With adults in the nest, fraction of surviving eclosion. So with adults in the nest, they all survived. Initially isolated and then returned to the adults in the nest, they mostly survived. Isolated in the tube, mostly survived, but that is with the fluid removed. When the fluid is not removed, they almost all die. However, ant nests are not clean environments. When pupae were isolated in vacant, used nest boxes, the fluid droplets became contaminated with fungi. And these infections spread and ultimately killed all pupae. Pupae were isolated until fluid droplets appeared and then returned to the nest with a group of adults showed high survival. This shows that in the colony context, pupae depend on adults to remove the secretion and would otherwise die of fungal infections. Ant pupae are typically considered to be inactive members of the colony, but our experiments show that in O. bayroi, pupae produce a type of social fluid that is consumed by the adults. This fluid is secreted in large volumes by the pupae during a specific window of development and elicits parental care behavior that is necessary for the pupal survival. So are they saying here that there's some sort of chemical in the fluid that prompts the adult ants to take care of the baby ants? Social fluid is derived from molting fluid. To understand the molecular composition of the pupal social fluid, we collected samples from a population of pupae reared in isolation and conducted proteomic and metabolomic profiling. To verify that the detected compounds were secreted with the fluid rather than stemming from contamination acquired via contact with the surface of the pupae, we compared pupal fluid profiles with profiles obtained from the pupal whole body washes. We collected samples daily during the 5 days before the pupae closed and analyzed them using liquid chromatography, tandem mass spectrometry. Hierarchical clustering of proteomic profiles revealed a clear distinction between pupil fluid and pupil whole body wash. Figure 2A. So this is protein abundance. Okay, so the pupil social fluid has a lot more protein. Interesting. Out of 212 identified proteins, 185 were either found exclusively or significantly enriched in pupil fluid compared to with whole pupil body wash. Proteomic gene ontology enrichment analysis identified the process of protein and chitin degradation, which are represented by functions of such as peptidase and chitinase activity, respectively. There's some words I do not know here. Proteomic gene ontology. I want to Google this here. Okay, it's all scholarly articles. What is this? Okay. I actually don't know what the word ontology means. I feel like that's pretty commonly used.
Ontology
is a branch of metaphysics dealing with nature of being. That is crazy. I feel like that makes sense with how I've heard it used. So gene ontology is trying to just understand how genes function, I guess. Defines the universe of possible functions a gene might have. Wow. That's cool. Okay, and this is referencing figure 2B. Metabolic intensity. Activities related to protein and chitin degradation are involved in the degradation of the old cuticle, which is one of 3 major molecular pathways characteristic of insect molting fluids, as has been demonstrated by proteomic profiling and functional analysis of Manduca sexta, Bombyx mori, and Tribolium castellanum molting fluids. I'm assuming those are all other species of ants. The other 2 major pathways found in molting fluids are immune response and regulation of protease activity. Gene ontology terms associated with both pathways are enriched in pupil fluid. The immune response pathway includes enzymes involved in cuticle melaninization, such as phenoletoxidase, consistent with our observation that the onset melanization always precedes pupil secretion. This is a key enzyme in the melanization cascade which participates in cuticle sclerosation, and wound healing. Interesting. So, does that mean when the ants are getting darker in color, they're developing their immune system? Seems like it, perhaps. And figure 1C. Figure 1C. Yeah, okay. We already looked at that. Molting fluids contain proteolytic and chitinolytic enzymes that catalyze the breakdown of chitin into free amino acids and N-acetyl D-glucosamine to provide additional evidence that the pupil fluid is derived from the molting fluid. We screened for these breakdown products. Targeted metabolic profiling of pupil fluid and pupil whole body wash indicated that the social fluid contains a variety of micro and macronutrients. These include all essential amino acids, multiple carbohydrates including that chemical I just mispronounced, nucleic acids, and vitamins. Figure 2C. Oh, we already saw that. Dang, my bad. Out of 107 metabolites identified, 105 were significantly enriched in the pupil fluid compared to the pupil whole body wash. Time series analysis revealed an increase in many free amino acids and glycnac as pupil approached eclosion. Moreover, glycnac was among the metabolites with the largest fold change. This is consistent with the findings from other insects where these metabolites increase in the molting fluid during the molting process. Fold change. That is something that I'm not familiar with. So these are all these nutrients and vitamins that are in the fluid here. Huh. This is consistent with the findings from other insects where these metabolites increase in the molting fluid during the molting process. These results show that the pupil social fluid is rich in a variety of proteins and metabolites that has the molecular and physiological characteristics of insect molting fluids. Non-eusocial Ecdysozoa reabsorb the molting fluid before ecdysis recycle nutrients. What is this? Ecdysozoa. Ecdysozoa. Ecdysozoa is a group of protostome animals. Oh, it's just a group of animals with exoskeleton. So I think it's just the group that like insects and worms and spiders are in. Cool. By contrast, clonal raider ant pupae secrete the larger proportion of the molting fluid from the pupal case, where it is consumed by adult nestmates. Young larvae consume the social fluid. Given the nutritive content of the pupal social fluid, we hypothesized that larvae which grow and therefore require the most food might also consume it. Colonies of Oviroi undergo phasic cycles that alternate between brood care and reproductive phases. Figure 3a. Did we not look at that? Oh, that's cool. I wonder if I could make this bigger somehow. Reproductive phase. Interesting. Cool. During the brood care phase, colonies contain late instar larvae that induce workers to leave the nest and forage for food. During the reproductive phase, colonies contain pupae, foraging activity ceases, and workers synchronously lay a new batch of eggs that hatch into the larvae at the end of their reproductive phase. Therefore, the brood develops in discrete cohorts, and each new cohort from the larvae hatches while the previous cohort is at the late pupal stage. Thus, the pupal fluid might provide an important food source for the larvae before workers begin foraging. Huh. To test whether larvae are present during this time in the colony cycle when pupae secrete fluid, we isolated pupae and eggs from colonies in the middle of the reproductive phase and daily recorded the number of pupae secreting and the number of larvae hatching. The first larvae hatched only 1 day before the pupae began secreting, showing that the two events are tightly synchronized. Larvae hatched, pupae secreting. Okay, so this is all timed out, and they all reproduce as a group. They all clone themselves as a group. We then placed larvae in a colony with pupae injected with food dye to test whether the larvae consumed the dye-strained pupal fluid. We consistently observed dye straining in larvae guts within 24 hours of the experiment. In the colony context, young larvae are usually attached to pupae, which often with their mouth parts suggested that they consume fluid directly as it appears on the pupal surface. Wow. And so probably saw that, but wow. So this guy is excreting the fluid and this guy's attached to him. So they just lay their eggs on top of the larvae that is currently growing. I wonder if it's the same ants that laid those larvae are just stacking them. Larvae have limited mobility and thus are dependent on adults to place them on food sources. Observations suggest that Oviroi adults readily place young larvae on pupae and older larvae on prey items. Older larvae on prey items. Oh, so like food that they brought in. To test the adults' preference for alternative food sources for young larvae, we provided colonies of 10 adults and 10 newly hatched larvae with either 10 melanized Obiroid pupae, both 10 melanized Obi pupae, and prey items. Dead pupae of the fire ant, Solenopsis invicta. That's such a cool name for an ant, Solenopsis invicta, and replaced every other day. So these ants, the Obiroi, are predator ants. They're raider ants. They raid other ant nests. So these fire ants here would be something that they would eat. And let's look up a picture of the Obiroi. Maybe we can, because I don't know. I want to see what they look like as adults. We've been looking at a lot of infant pictures of them. Wow. That's cool. Or 10 prey items alone. We then recorded the location of the larvae once a day for 5 days. When given pupae alone, ants placed almost all the larvae on pupae until eclosion. When given a choice, ants showed a strong preference for placing larvae on pupae as opposed to the prey. Even in absence of pupae, ants did not place the young larvae on the prey. Thus, ants place newly hatched larvae and early instar larvae on their own pupae where they have the ability to feed on pupal fluid, and only place later instar larvae on prey, and only later place instar larvae on prey. To determine whether this relationship contributes to larval growth and survival, we conduct an experiment in which larvae hatched in social isolation at 0 to 12 hours of age without prior access to food were fostered into colonies of adult ants. Colonies were composed of 10 adults and 5 fostered larvae each, and were supplied with either 10 melanized Obiroi pupae, 10 prey items, dead S. invicta pupae, replaced every other day, or neither. We measured larval growth and survival over 4 days after hatching. Larvae colonies with pupae grew significantly more and had significantly higher survival compared with the larvae in colonies without pupae, regardless of whether the prey was present or not. Let's look at these figures for a second. Fraction of larvae on pupae. Days after hatching. So this is larva body length. And this is what they're eating. So there's a much, so the ants that are feeding off of the pupal fluid grow much larger and much faster. Fraction of larvae surviving. Yeah, and is much more likely to survive when they're feeding off the pupal fluid. All right, where is I think it is right here. Okay. All pupae in these colonies enclosed. All pupae in the colonies closed, showing that pupae themselves were not consumed. Brood cannibalism is a commonly observed response to stressful conditions in ant colonies. There were no significant differences in growth and survival between larvae in colonies that were given prey and those kept without prey. Thus, we observed faster growth and higher survival of larvae reared in the presence of pupae, and found no evidence for a similar beneficial effect of prey items. These experiments show that the pupal fluid serves as a milk-like substance for newly hatched larvae, greatly increasing larval growth and survival during the first days after hatching. Together, our results reveal that the pupal molting fluid has previously unknown yet important role in O. baroi's social organization and colony fitness. Ooh. Pupal secretions are conserved across ants. Despite its importance in Ossuae and Oviroi ant subfamily Dorylaenae, to our knowledge, a social function of pupal secretions as trophylactic fluids has not been described in any of the 14,000 ant species. To test whether this function is specific to Oberoi, we socially isolated pupae from 4 other species to cover the 5 major ant subfamilies: S. invicta, Nylandaria flavipes, Tapenoma sessile, and Ponera pennsylvanica. Pennsylvanica. We found that melanized pupae of all 4 species secrete fluid droplets from the abdominal tip. Metabolomic profiling showed that the composition of these secretions is similar to that of Oberoi. Out of the 70 metabolites identified in Oberoi in this analysis, we found 69 in at least 1 other ant species, including free amino acids and glycnec. This shows that across the ants, pupae secrete a liquid derived from molting fluid. To test whether pupal fluids also have a social role in other ant species, we injected blue food dye into the exuvial space of melanized S. invicta and placed them into small colony fragments. After 24 hours, the digestive systems of all adults and most early instar larvae were stained blue, indicating that they had ingested pupal fluid. As previously showed that S. invicta did not enclose in social isolation, and concluded that adults were required to help remove the exuvia during eclosion. By contrast, while pupae died if their secretion was not removed, manual removal of the social fluid was sufficient to achieve high survival rates without any additional assistance during eclosion. Together, our results show that secretion of molting fluid by pupae is widely conserved in ants, and that dependence on pupae on other colony members to consume the fluid is not limited to Oberoi. In many ant species, adults place young larvae on pupae and that adults drink dyed pupil fluids is directly visible in N. flavipes because the abdominal of these ants is partly translucent. In ant species whose pupae are enclosed in cocoons, such as P. pennsylvanica, adults consume social fluid through the permeable silken fabric. This can be visualized using dyes in ants with translucent cuticles, such as Myrmecocystis mexicanus. Additional work will be required to study the social role of pupil fluid across the ant tree of life. Although the social functions of the pupil secretion must be a derived ant trait, it remains to be determined whether pupae of solitary hymenopterans produce secretions derived from the molting fluid. What is this word? Hymenopterans. Hymenopterans is a large order of insects comprising of bees and ants. Cool. So I guess bees and ants are kind of like cousins, maybe. If so, these secretions might have readily been co-opted during the various independent evolutionary origins of Hymenopteran eusociality. We examined the possibility by socially isolating honeybee, pupae. None of these pupae produce visible secretion droplets. And more than 80% of the pupae survived to eclosion in social isolation without additional assistance. Although this shows that not all eusocial clades rely on social functions of pupil fluids, the evolutionary dynamics of pupil secretion across Hymenoptera will be a rewarding avenue for future investigation. So bees do not share this trait of ants. During major evolutionary transitions, initially independent units become integrated and mutually dependent to form higher levels of biological organization. Here we show that ant pupil molting fluid has acquired novel social functions that create previously unrecognized interdependence between pupae, larvae, and adults. We have demonstrated that this fluid is detrimental for pupae if not removed, and that it is an important food source for early larvae. This secretion is likely to have additional far-reaching effects on larvae and other colony members. Previous work has shown that trophylactic fluid from adults contain not only pre-digested food, but also endogenous molecules such as juvenile hormone, a known regulator of insect development and behavior. Similarly, the pupil fluid described here contains hormones and neuroactive substances that may modulate the development and behavior of larvae and adults. Far from being passive colony members, pupae thus have an active and central role in ant colony organization. Nice. Online content. Any methods or additional... Okay, I don't think we need to read that. Wow. So I don't think we need to go into super in-depth into their methods. But I do want to read their conclusion if they have a conclusion. Whoa. We should look at some of these pictures though. So this looks like probably where the fluid is coming out here. 1A and B. Scanning electron microscopy images of young, 1 day old. Wow. Yeah. So yeah, that's where the fluid comes out. What is C? Is that the beginning of the droplet of the tropophylactic fluid? Yup. Yup. Fraction of adults with staining. What is F referring to? Adults in Oberoi showing staining of the digestive system after spending 24 hours of pupae that were injected with a food dye, right, but not when the food dye is offered in a cup in the same amount. Okay, so they only eat it off of the larvae. If you put the same fluid in a cup, they do not. Oh no, this is just the dye. So they were just making sure that the ants were not attracted to the dye. They were attracted to the dyed fluid. Wow, so this is a full list of all the different nutrients. It's amazing how, like, in something so microscopic will just contain all these different chemicals, you know? It's honestly crazy. So I saw this image and I'm not sure what this is. It might be the gene ontology analysis that they were talking about? Yep, that's what it is. Nodes correspond to enriched geo terms. I feel like I don't think this is like an exact science. I think this is kind of just like, I really don't understand, I guess, how they figure out this information. But I guess so. These are just things that are associated with this fluid. Chitin-based cuticle development, male genitalia, morphogenesis. And so this is in the body wash. And so still quite a bit of, oh wait, no, never mind. Okay, some of these are old figures just showing it higher, closer up, which is really nice. Oh my gosh. Interesting. So this is them placing the larvae on the pupal ants. Wow. What is A? Scanning electron image of the cocoon. It illustrates a porous fabric. Wow. So yeah, so even in this other species of ants that where they form inside of a cocoon, they still find a way to get that pupal fluid out of there. Jeez, look at that. These things look so alien. But they're just like us. We also, actually no, we don't eat any fluid from. I'm just going to stop talking. Wow. Okay, so that is the article. And in summary, ants are utilizing the fluid from their larvae. They're feeding it to they're eating it themselves. They're feeding it to their even younger larvae, um, and it's the fluid produced by their young is very important to way that they the way that they develop. It's important to their immune system, and it might actually also induce like a behavioral change in them. So this is pretty cool to learn about. So yeah, I'm gonna sign off here.